If the doors of perception were cleansed, everything would appear to man as if it is, infinite. This is arguably the most widely known quote from the famous book titled The Doors of Perception by the English writer and philosopher Aldous Huxley. At the time, it was 1954, and the discovery of DNA was only a year old. Consequently, little was known about the genetic foundation of serotonin and dopamine receptors in the brain, and their role in such an experience described in the book. For those of you who are not aware of this infamous book, it is a vivid outline of an experience that Dr. Huxley had immediately prior to taking nearly half a gram of mescaline, which is a hallucinogenic drug, dissolved in about half a glass of water. Mescaline is known to bind to serotonin and dopamine receptors in the brain, including the hallucinogenic properties that we all know and love. Other such drugs, the Big Five as I like to call them, include lysergic acid diethylamide, commonly referred to as LSD, psilocybin and muscarine, which are found in mushrooms, dimethyltryptamine, which is found in our brains and is released in our dreams, and 3,4-methylene dioxymethamphetamine, commonly known as MDMA. All of these drugs share the same physical story. Once they enter our system, they target the receptor, or a receptor, and then work their magic. What this magic is, though, we wish to uncover. So you might be wondering why I am starting such a series on the animated physics of drugs here. First, why am I calling this the physics of metaphysics? Both terms seem to mutually be exclusive, as we typically understand physics to describe things that we can observe in the real material world, while metaphysics operates within the domain of a more abstract and non-material world. Physics concerns itself with calculating the gravitational pull of two large bodies or the quantum properties of chemicals, while metaphysics, on the other hand, deals more with the rules and ethics and value. But that's the thing, value. We as material human beings observe the world, and during this process, we attribute value to the various things that we see around us. One could ask then, what causes the nervous system to value things more than others? It is my belief that drugs offer us an answer and will act as a gateway to the so-called non-material world. This is the world of the gods, value and morality, instead of the world of people, objects, and interactions. But it is also my belief that such drugs mediate how we experience the metaphysical world as well. Also, by the way, my notes are underway. I have decided to do something very special with my notes that you'll be able to take a part of in their growth. I'll be releasing them slowly. I will update them on a weekly basis, where if you contribute to a new tier that I'll be creating within the next few days or so, you will have access to all of them. And then a higher tier will give you the ability to not just access them, but contribute to them and be a co-author in them as well. This tier is more expensive than the current tier, or these two tiers will be more expensive than the current tier I have right now. But they'll constantly be updating, and you will have access to all of them. So I think this is a good deal for anyone who decides to take it. An extra video also is going to be coming out on how you will be able to access these notes within the next few days. Anyways, I digress. Let's get back to the content of the video. Well, here I am in the lab, slavishly working away at setting up a purification for a type of glutamate receptor. This is the kind of stuff that I really enjoy. As much as I love to learn and teach advanced physics, I also have a very strong graduate background in biophysics. I've always been interested in integrating physics with the life sciences in an attempt to revolutionize how we do healthcare and education. Well, I'm not quite there on the healthcare front, but I like to think I'm there with the education front. Anyways, these receptors are sensitive to the neurotransmitter glutamate, but will bind to other, perhaps more widely known drugs, such as ketamine. It's going to turn out that there is a rich and vast underlying physics that dictates how such drugs bind and affect these receptors. And it's because of this physics that we get the side effects that we do. My intention with this new video series, and these videos to come on this topic, is to bring to light these drugs and bring them to the top of our awareness. While they might be effective, these drugs are no laughing matter. Understanding their fundamental mechanism of action will help the general public become more aware of how dangerous these drugs really are, and who knows, it might bring one of you listeners to want to pursue addressing the problem that these drugs like fentanyl and ketamine pose. I want to take this moment also to mention a few things about my motivation behind making this series of videos. I quickly want to also dedicate this video to my cousin. He was also my best man during my wedding. A few months ago, he passed away while playing an innocent game of basketball with his friends. He went into cardiac arrest after the game, and within 24 hours he was gone. This was a devastating blow to me, my family, and his family because he was only 28 years old. The cause of his passing was a rare genetic cardiac mutation in his heart called Brugada syndrome. This syndrome is quite rare and is a consequence of a mutation in the sodium channels that are responsible for keeping the heart beating. This quote is a quote that he gave during my wedding. The genius of communication is the ability to be both totally honest and totally kind at the same time. This video is dedicated to him. I feel like I kind of have to explain myself in my absence over the past few days also. Along with this series, I'm going to be producing more on string theory, twistor theory, spinors, quantum field theory, general relativity, and all the rest that I've already been covering, but now in an animated way. Over the past few weeks, I've been working on animations with Python, Blender, Adobe After Effects, Samsung 1 Angstrom, and Manum to develop a seamless workflow to produce high-quality videos at a relatively fast rate with awesome looking animations. If you support this channel, these videos will get produced and published faster. Now let's get to the topic. Well, so my cousin was the primary reason I decided I wanted to start this new series of animated videos on biophysics. The second reason, though, I've decided to start making this series is because I recently watched the Jordan Peterson Jubilee debate. After watching the debate, I began listening to a lot of commentary afterwards, and the commentary was sort of a mixed bag, with some people pointing out the inconsistencies of atheists, and other people pointing out the haphazard manner by which Dr. Peterson defended and claimed to be a Christian. Well, the thing about Christianity is, and I might get some pushback on this, but it's true, is that a large part of it was founded on the use of plants. The Old Testament, for example, mentions shittim wood 52 times. The altered states of consciousness that Adam and Eve achieve in the Garden of Eden came from eating from the tree of knowledge. There are books written about the influence that plants had on the development of Christianity. This isn't to say that Christianity was founded on just tripping on plants. Rather, the point that I want to get across here is that Christianity, along with many other cultural traditions, discovered the world of the spiritual through plants, and these plants sort of acted as a gateway to this discovery. So the natural question is, how do plants work on our physiological systems in order for us to reach the pearly gates, so to speak, without having to undergo the process of death? Well, that is what this series is going to uncover. Okay, let's start with a simple overview of the nervous system. The nervous system is a complex network of cells within our body that communicate with each other. This communication, however, is mediated by synapses, and roughly speaking, each synapse calculates how an electrical signal is partitioned through the body. If we look at this animation, for example, and we focus on just one area, we see an electrical pulse generated by the brain and going to the feet. But as it goes to the feet, there's a crossroads. There's another major nerve cell along the way, and depending on the person's physiological and mental state, the synapses at this node will determine the amount of signaling that's allowed to pass into the secondary nerve cell. Let's look at this in a little bit more detail. We can see in this animation that I've generated here that we have vesicles containing neurotransmitter, okay? A pulse is generated, and then calcium is going to flow into the cell. And then once that calcium enters the cell, that's going to allow for these vesicles to then bind, open or release their neurotransmitter into the synapse. These neurotransmitters bind to more receptors on the receiving end, which then open and allow for sodium ions to pass through, as we've seen. Okay, this is the general story for most cells, right? What's going to be nuanced here is the physics of the receptors, and that's what we're going to look at in this series. And so there you have it. The electrical signal causes vesicles to travel down the axon and towards the synapse. And this is where the calculation happens, at the synapse. There are a lot of different types of receptors, though, that are ready to latch onto their associated ligand. And once they do, the receptors open and allow for the flux of ions inside the receiving cell. Fundamentally, what's happening here is a transfer of electrical signal to chemical signal, where the electrical signal propagates and the chemical signal essentially calculates. But this is usually where the story stops for those of you who are taking a course in medical school, or if you're an undergraduate student. If you're a listener of Artem Kirsanov, which I proudly am, then you might go a little bit deeper into what the implications are from a more neuron level perspective, because he's a neuroscientist and that's what he does, and it's very, very interesting what he does. However, I'm a biophysicist, and while most of his work in his recent video was great, he left out the whole world of physics that is involved in uncovering these receptors and how they actually work. And that's what we are going to continue doing in this video. The reality of the situation is that there is a whole host of new physics that's actually found at the molecular level here. Embedded within the synapses are hundreds of different types of receptors, as I've mentioned, that are all susceptible to various different types of neurotransmitters and drugs that you take. The neurotransmitters don't just bind to receptors and cause them to open, rather, the neurotransmitter actually modulates the probability of the receptors being open. These receptors have a stochastic nature to them, and so will be opening and closing all the time. Drugs and neurotransmitters just change their open probabilities. They also change their amplitude of activation, rates of desensitization, and rates of deactivation, which are all concepts that we're going to go into soon. To get at the physics, though, we need to be able to find what is common between all of these receptors. Well, one way to look at the receptors is to coarse grain out all the detail of the atoms and just look at the domains of the protein. Generally speaking, most receptors that are membrane bound to the synapse will have three domains. These are the C-terminal domain, the transmembrane domain, and the N-terminal domain. The formation of these domains comes from the fact that amino acid strands will contain certain local regions that will interact with each other. The result is a protein with different domains. This is what is commonly referred to as the tertiary structure of the protein. The primary and secondary structures are just a sequence of the amino acids and the formation of the alpha helices and beta sheets. After this process is done, a quaternary structure is formed. This is when multiple different tertiary structures begin to interact with each other. Whenever you hear the term tetramer, for example, this means that 4 strands of amino acids formed the quaternary structure. This is all very preliminary, but the point that I want to get across here is the idea that proteins can be looked at in a more coarse-grained way. Instead of considering every single atom and how they contribute to the overall function of the protein, we just look at distinct regions of the protein and study how they modulate the overall function. The end result is a tiny little machine with different moving parts. Instead of looking at the atoms of a machine, we look at the parts of the machine. Parts that are not able to form distinct regions are typically not studied very much because they're so flexible, so researchers will tend to omit them in their publications. Nonetheless, they are still very important. In kinase receptors, for example, the phosphorylation of the unstructured C-terminal domain is important in contributing to seizures. So this is a picture that we're going to be looking at now for most of what's to come. For most ligand-gated ion channels, that's channels that open when they bind to some small molecule like LSD or fentanyl or ketamine or heroin. Within their domains that stick outside of the cell, they contain essential subdomains for proper functionality. These are their N-terminal domains, or NTDs, their agonist binding domains, or ABDs, their transmembrane domain, or TMDs, and their C-terminal domain, CTDs, all of which contribute to the overall functionality of the protein receptor. So what are we going to do now with this picture? Well, we aim to identify certain states of the protein, and this is going to require an understanding of protein kinetics. And depending on how ambitious I am, and I'm feeling pretty ambitious here, we're going to dive into some crazy stuff on statistical mechanics. So what is protein kinetics? Well, we know that a general protein can be closed or open. The closing or opening of a protein is determined by knowing if we can measure a current or not. If we can measure an electrical current, then we know that the protein is open. And for illustrative purposes, I've overemphasized the degree of opening of the channel here. In the context of a synapse, an open protein is one that is bound to neurotransmitter. So we could construct a simple model for protein states. One node in this model is the node that represents the closed or non-conducting state of the protein, and the other is the open or the conducting state of the protein. We connect two nodes with arrows and assign a variable to each arrow, and each variable is going to represent the rate of the protein transition between the two states. So we have a rate of closure and a rate of opening. Furthermore, we can also attribute a probability to each state. The physics of these drugs lies in the fact that these numbers will be different depending on the drug, and the probability of the closed state is just 1 minus the probability of the open state, because we're just looking at two states right now. We know, for example, that fentanyl, a very dangerous and extremely lethal drug, targets serotonin receptors. They are an agonist, meaning that they will cause the receptor to favor the open state, so the probability of the protein being in the open state increases, while the probability of the protein being in the closed state decreases under the influence of fentanyl. This does not necessarily mean that the rate of going to the open state increases, though. But there is a relationship between P open, K open, and K closed. We can actually use the following equation to generate some Python code that independently modulates the rate of closure, the rate of opening, and the open probability. Probabilities have an upper bound, whereas rates of transition do not. If the probability of seeing a protein in the open state increases, then we might infer that the rate of transitioning from the closed state to the open state increases as well. However, that's not necessarily the case, and this could mean that the rate of going from the open state to the closed state actually decreases. So either the closed state becomes less stable under the effects of fentanyl, or the open state becomes more stable. Now here's where we start getting into some interesting physics. These three constants are related in the following way for our system in equilibrium. And this equation makes sense to some degree. If you are more likely to see an open state protein, then the fraction here on the right side of the equation is greater than 1, and so K open is larger than K closed. If we multiply both sides by the denominator, we get an interesting conservation law. We will come to understand this as something of a probability flux, if you will. You take the probability of a node and multiply it by the incoming rates, and the value that you get is the same value across all nodes. And this will apply to higher order graphs as well, as we'll see in later episodes within this playlist. And this is a perfect transition now into the idea of multiple node graphs. In reality, proteins have more than two states. Some proteins, for example, can be semi-open, AMPA receptors have the ability to have multiple open states where each one has a different current amplitude, and each one is defined as being bound to 1, 2, 3, or 4 ligands, or 4 small molecules. We'll see how this works later. Some proteins can also have multiple closed states. NMDA receptors can be closed but also be plugged up by a magnesium ion, both of which are different in their relative energies. So each state can be uniquely characterized structurally, which is what the whole enterprise of structural biology is, but can also be characterized or uniquely characterized energetically, which is what the whole enterprise of statistical and non-equilibrium thermodynamics entails, all of which we're going to cover in this animated series. But for right now, I don't want to overwhelm you with too much information. I want to swing back to when I mentioned Christianity and the Jubilee debate that Jordan Peterson had on recently. This debate was frustrating, for lack of a better term, but there was a particular part in the debate that stood out to me. For copyright purposes, I'm not going to play the video, but the young gentleman who confronted Peterson noted that, quote, God says that you can own people as property and beat them. He then goes on to ask Peterson if this was, quote, in line with Christian ethics. And my response is this. This clip occurred around the 1-hour timestamp of the debate. Yes, God is described as having commanded many bad things in the text, but there are many different examples of vengeful gods in different cultures, suggesting that vengeance and bloodshed isn't unique to the Christian God, but is more reminiscent of something akin to the vengeance of human nature and tribalism. Such commandments given by God have been suggested to be a product of a more bicameral and more primitive mind, as outlined in the book The Origin of Consciousness and the Breakdown of the Bicameral Mind by Julian Jaynes. Roughly speaking, this would be a mind that takes in sensory information and just responds. The sensory information being commands from God, and the response being doing what God says. The concept of thinking was an evolved process, in other words. My broader point here is that the ancient mind contained the same serotonin receptors, the same glutamate receptors, the same GABA receptors, and so forth. So, what exactly changed in our brains to push these receptors into making the brain a more inquisitive and more thoughtful one? The gist of what I'm going to argue is that pointwise mutations in these receptors will change the rate by which these receptors open and close, just as we've seen LSD, psilocybin, and other drugs do in this video to our kinetic models. Environmental conditions, as we know, through the process of evolution, will select for the phenotypes of these pointwise mutations, ultimately changing how the brain responds to external stimuli. Less reaction time to external stimuli will correspond to the development of a mind that thinks more and responds less. The attempt of humans to go back to the state where gods would just tell them what to do would be harder and harder as evolutionary time progresses forward. I want to make clear that I do believe in God here, but I try to be as biological and physical and concrete as possible with my arguments, as uncomfortable as they might be. This series is going to be a comprehensive video on statistical physics, biophysics, and so much more, and I think that are all going to point towards the strongest argument for God that I have managed to formulate here. Also, make sure to hit that like and subscribe button, and also if you contribute to the channel, not only will you be doing yourself a favor, but also humanity a favor as well. Bye.